Welcome to the PubMed Daily Review for May 25th, 2026. Today we scanned 976 new open access papers across 383 journals. Here are the top 10 selected for detailed analysis. Trend 1 of 10, diverse clinical medical research. This trend encompasses varied clinical studies exploring disease, treatment, and patient outcomes across different medical domains and methodologies. This cluster covers 118 papers. Representative work includes machine understanding, traveling with HIV, and geography as a determinant of lung transplant access in the United States. Trend 2 of 10, developmental and reproductive cell biology, investigating cellular mechanisms and factors influencing developmental processes and reproductive health across various biological systems. This cluster covers 39 papers. Representative work includes microglial ontogeny and in vitro reconstruction, bridging development and modeling, ALDH1A3, dependent ferroptosis mediates hexafluoropropylene oxide dimer, acid-induced trophoblast dysfunction, and decidualization potential of endometrial mesenchymal stem cells and their role in reproductive health. Trend 3 of 10, cancer genomics and clinical studies. Integrating genomic insights with clinical research to advance cancer understanding, diagnosis, and treatment. This cluster covers 75 papers. Representative work includes improving physical activity in men with prostate cancer through wearable devices and online education. Randomized controlled trial. Molecular and spatial differences in the tumor microenvironment of high-grade serous ovarian cancers with short versus long-term survival. And enhanced recovery of high-quality DNA from limited FFPE tissue for advancing cancer genomics. Trend 4 of 10, microbial genomics and applied biology. Focuses on leveraging genomic data from microbes and related biological systems for diverse applied biological solutions and understanding. This cluster covers 122 papers. Representative work includes preclinical dental students' awareness and integration of pulp biology knowledge, a cross-sectional study assessing the understanding of pulpal functions and implications for clinical endodontic competency and practice. Chromosome-scale genome of cheilicoids brevitarsis provides a resource for comparative and vector biology studies, and primary challenges in the upcycling of genetically modified spent microbial biomass. Trend 5 of 10, neuroscience and mental health studies. This trend explores neurological bases, computational analyses, and clinical manifestations of mental health and neurodevelopmental conditions. This cluster covers 93 papers. Representative work includes large language model approach to uncover reasoning patterns in forensic psychiatric assessment, investigation of phubbing behavior in attention deficit hyperactivity disorder and cognitive disengagement syndrome, a case control study in adolescent, and free water, occipital volume, and changes in depressive symptoms in the Life After 90 study. Trend 6 of 10, nutrition, public health, and metabolism. Investigating the intricate links between nutrition, metabolic health, and public health outcomes across diverse populations and diseases. This cluster covers 100 papers. Representative work includes factors associated with malnutrition among under 5 children in Bangladesh, a multivariate analysis, systemic lipid abnormalities and choriocapillaris flow deficits in patients with hard drusen, and impact of emphysema on biomarkers in hospitalized COPD patients with community-acquired pneumonia, a retrospective study. Trend 7 of 10, clinical trials and treatment efficacy. Studies evaluating diagnostic methods, treatment efficacy, and disease characteristics using clinical trials and systematic reviews. This cluster covers 101 papers. Representative work includes automated measurement of the minimum macular hole diameter based on optical coherence tomography images, effective preoperative oral Montelukast prescription on postoperative pain levels following zygomaticomaxillary complex fracture surgery, a randomized control trial, and prevalence of xerostomia and hyposalivation in older adults. A systematic review and meta-analysis. Trend 8 of 10, health education and public engagement. This trend explores educating individuals and engaging communities in health-related topics, research, prevention, and professional development. This cluster covers 99 papers. Representative work includes democratizing health and social care research through long-term public involvement and engagement, a qualitative process evaluation of the Community Research and Engagement Network, COREN, spotlight on mentoring undergraduate medical students using evidence-based medicine and the evolving landscape of primary headache therapy. Trend 9 of 10, AI and deep learning applications. Applying AI and deep learning techniques to solve complex problems across diverse scientific and engineering domains. This cluster covers 110 papers. Representative work includes robust joint optical delay and received signal strength positioning for visible light communication systems using particle swarm optimization, geometry-aware localization evaluation of grad-CM for wafer map defect classification, and flashover prediction of polluted composite insulators based on arc time constant and velocity using CNN LSTM. Trend 10 of 10, molecular mechanisms and drug discovery. Unraveling molecular mechanisms underlying biological processes and diseases to inform and advance drug discovery efforts. This cluster covers 119 papers. Representative work includes activation of the RAS/PAR pathway reduces back fat deposition of GALE, bos frontalis. Synkinase I inhibits uric acid reabsorption by binding to the TRP459 residue of the GLUT9 protein, and targeting EPCM expression via near-infrared fluorescent antibodies enables microscopic delineation of primary and recurrent HNSCC. Up next, our first paper out of 10, Breaking Barriers Between Species. Integrated multi-species PK models improve human PK predictions by Miriam S. R. Happ and Bettina Gerner from Research DMPK. Global Drug Discovery Sciences, Boehringer Ingelheim Pharma GmbH & Company. KG, Ingelheim am Rhein, Germany. This paper was selected because it introduces integrated multi-species pharmacokinetic models that significantly improve the accuracy of human pharmacokinetic predictions, directly addressing a critical and costly bottleneck in drug discovery and development. Let's break down its key contributions. Predicting how drugs behave in the human body, or their pharmacokinetics, is absolutely crucial in drug discovery. It helps us pick the best compounds and figure out the right dose. However, accurately predicting specific parameters that define the shape of a drug's concentration over time, like absorption and distribution rates, has been a real challenge. This research tackled that gap, aiming to make those predictions much more precise. Our team developed an innovative integrated multi-species PK model framework. This new approach combined allometric scaling with established mechanism-based prediction methods for bioavailability and clearance. We tested its performance against a benchmark literature framework using a dataset of 40 small molecules with known human PK profiles. The goal was to see if our new model could more accurately predict the complex shape of drug concentration curves. Our results were very encouraging. The new predictions showed a significant reduction in the geometric mean fold error, indicating much greater accuracy. Specifically, we saw improved prediction of intercompartmental clearance, Q, and late concentrations, C24H, which are critical for understanding drug exposure. This improvement was primarily driven by compounds that previously posed challenges due to inconsistent PK model structures across species, while performance for other compounds remained comparable. This study offers a significant step forward in drug discovery. Our integrated multi-species PK model provides a more robust and accurate way to predict human pharmacokinetics, particularly for challenging compounds and for predicting trough concentrations, which are crucial for efficacy and safety. By improving these predictions, we can make better decisions earlier in the drug development process, potentially leading to safer and more effective medicines reaching patients faster. This work paves the way for more efficient and reliable drug candidate selection. Up next, our second paper out of 10. CYP8B1 suppressed by RAS/ERK leads to cholesterol accumulation and bile acids reduction and facilitates hepatic tumorigenesis by Wan Li and Jun Shun from Laboratory Animal Center, Dalian Medical University, Dalian, Liaoning, PR China. This paper was selected as the top choice because it elucidates a novel and critical molecular mechanism involving CYP8B1, cholesterol/bile acid homeostasis, and RAS/ERK signaling that directly contributes to. Let's break down its key contributions. Hepatocellular carcinoma, or HCC, is a serious liver cancer, and we know that the balance of cholesterol and bile acids plays a crucial role in its development. However, the exact mechanisms, especially involving a key enzyme called CYP8B1, haven't been fully understood. This research aimed to shed light on how CYP8B1 and cholesterol/bile acid homeostasis contribute to HCC, particularly when driven by aberrant RAS/ERK signaling. To tackle this complex question, the researchers employed a comprehensive approach. They analyzed clinical data from TCGA and patient samples, conducted experiments using various cell lines, and utilized an ROS12V transgenic mouse model of HCC. Their methods included detailed molecular analyses, histopathology, and interventions like an ERK inhibitor and dietary modifications, allowing them to investigate the intricate interplay between RAS/ERK, CYP8B1, and cholesterol/bile acid balance in HCC. The study revealed several critical findings. They observed frequent ERK hyperactivation, reduced CYP8B1, and cholesterol accumulation in both clinical HCC samples and cell lines. In their mouse model, cholesterol accumulated due to attenuated bile acid synthesis from reduced CYP8B1 activity. Importantly, inhibiting ERK activity with AZD6244 significantly elevated CYP8B1, increased bile acids, and reduced cholesterol levels. Furthermore, they found that SHREP2 plays a key role in this regulatory pathway, and remarkably, treatment with TDCA, a bile acid, significantly inhibited hepatic tumorigenesis and development. This research provides a novel theoretical foundation for understanding HCC. It highlights that the suppression of CYP8B1 by RAS/ERK signaling is a critical driver of tumor progression through its impact on cholesterol and bile acid balance. The finding that TDCA can inhibit tumorigenesis offers a promising therapeutic perspective. However, the study also revealed the nuanced role of cholesterol, where a high cholesterol diet suppressed progression but not initiation, suggesting that further research is needed to fully unravel these complexities and translate these insights into effective clinical treatments for HCC. Up next, our third paper out of 10, Post-Infectious Psychiatric Symptoms and Diagnosis in Children and Adolescents Following COVID-19 by Mehmet Singizi and Yasmin Taz-Toran from Department of Pediatrics, Özel Girlife Hospital, Akinsel Street, No. 1 Tepebisay, Eskişehir, Turkey. This paper was selected because it addresses a highly pressing and difficult challenge in global health by providing crucial real-world data on the post-infectious psychiatric sequelae of COVID-19 in children and adolescents. Let's break down its key contributions. The COVID-19 pandemic brought unprecedented challenges, especially for children and adolescents, sparking worries about their mental health. This study aimed to investigate the specific psychiatric symptoms that emerge after COVID-19 infection in young people. Identifying these symptoms and their associated risk factors is incredibly important. It helps us understand who might be most vulnerable and how we can best support them. Researchers conducted a follow-up study involving 107 children and adolescents, all of whom had a confirmed COVID-19 infection. They were screened using the Revised Child Anxiety and Depression Scale, or RCADS, and those with elevated scores received a detailed psychiatric evaluation based on DSM-5 criteria. The study aimed to identify psychiatric symptoms and diagnoses post-infection, also looking at how these symptoms related to various risk factors. This comprehensive approach helped paint a clear picture of the mental health landscape after COVID-19. The findings were quite striking. Over half, specifically 56.1% of the children and adolescents developed post-COVID psychiatric symptoms. Even more concerning, 52.2% of those with no prior psychiatric history developed new symptoms. The parent-reported anxiety scores showed excellent diagnostic accuracy with an AUC of 0.92, indicating its strong potential as a screening tool. Importantly, children without a prior psychiatric history had significantly lower scores, reinforcing the impact of COVID-19 on mental health. This study underscores the critical need to monitor the mental health of children and adolescents following COVID-19 infection. We now know that factors like prematurity and prior psychiatric history significantly increase the risk of developing new symptoms. The good news is that the RCAD screening tool proved highly effective, offering a valuable resource for early detection. This research paves the way for more targeted interventions and support systems to help our young people navigate the mental health aftermath of the pandemic. Up next, our fourth paper out of 10, Deep Medullary Vein Drainage and Unfavorable Outcomes in Stroke Patients After Endovascular Therapy, A retrospective study by Jiaoding Wei and Jinsheng Zheng from Department of Neurology, the first affiliated hospital of Sun Yat-sen University, Guangdong Provincial Key Laboratory of Diagnosis and Treatment of Major Neurological Diseases, National Key Clinical Department and Key Discipline of Neurology, Guangzhou, Guangdong, People's Republic of China. This paper was selected because it identifies a novel prognostic factor, deep medullary vein drainage, which significantly impacts outcomes in stroke patients undergoing endovascular therapy, addressing a critical challenge in managing a leading cause. Let's break down its key contributions. Today, we're looking at a study that explores a critical question in stroke care. Ischemic stroke, especially in deep brain regions, relies on proper drainage through deep medullary veins. But what happens if this drainage is disrupted after a procedure like endovascular thrombectomy? This paper investigates if such disruption could lead to worse patient outcomes, a crucial area for improving how we treat stroke. This was a retrospective observational study conducted across 2 stroke centers. Researchers included 61 ischemic stroke patients who had undergone successful endovascular thrombectomy for MCA or ICA occlusion. The key exposure they looked at was the continuity of deep medullary veins, assessed using susceptibility-weighted imaging after the procedure. The primary outcome was an unfavorable modified Rankin scale score at 90 days, comparing patients with and without DMV discontinuity. The study found that nearly half of the patients, 49.2%, experienced unfavorable outcomes. Critically, deep medullary vein discontinuity was much more common in this unfavorable group, at 70% compared to 41.9% in those with favorable outcomes. After adjusting for other factors, DMV discontinuity was independently associated with unfavorable outcomes, showing an adjusted odds ratio of 3.23 with a statistically significant p-value of 0.030. A combined predictive model, including DMV status, also demonstrated good prognostic value with an AUC of 0.81. This research highlights that impaired deep medullary vein drainage is a novel and significant predictor of poor outcomes after endovascular thrombectomy. Clinically, this suggests that assessing DMV continuity using SWI could become a valuable tool for risk stratifying patients after their procedure. However, it's important to remember this was a retrospective study with a relatively small sample size, so these findings will need to be validated in larger, prospective studies to confirm their generalizability and clinical utility. Up next, our 5th paper out of 10, Beneficial Roles of Selenium and Docosahexaenoic Acid Against Prenatal Methylmercury-Induced Adverse Effects on Child Neurodevelopment. The Tohoku Study of Child Development at 42 Months by Nozomi Tatsuda and Kunihiko Nakai from Health and Environmental Risk Division, National Institute for Environmental Studies, 16-2 Onagawa, Tsukuba, Ibaraki, 305 Japan. This paper was selected because it significantly advances public health understanding by demonstrating that beneficial nutrients in fish can mitigate the neurodevelopmental risks of prenatal methylmercury exposure, thereby refining critical dietary recommendations for pregnant. Let's break down its key contributions. We know that prenatal exposure to methylmercury, or MHG, can negatively impact a child's development, which often leads to advice for pregnant women to limit their fish intake. But this creates a dilemma, as fish is also a rich source of vital nutrients like DHA and selenium, both essential for healthy brain development. This study aimed to untangle this complex relationship by examining how combined prenatal exposures to MHG, DHA, selenium, and lead might influence the intelligence of young children. This study followed a cohort of 471 children from the Tohoku Study of Child Development. Researchers assessed prenatal exposure by measuring biomarkers like maternal hair mercury, maternal plasma DHA, and cord plasma selenium. The primary outcome was the children's intelligence quotient at 42 months, specifically focusing on simultaneous and mental processing scores using the KBC assessment. This design allowed for a detailed look at how different levels of these prenatal exposures correlated with early cognitive development. Our findings revealed some intriguing differential associations. In males only, maternal hair total mercury was significantly negatively associated with processing scores. Conversely, maternal plasma DHA showed a positive association with these same scores in males, and cord plasma selenium also had a marginal positive association. However, when using more complex models like quantile G computation and BCAMR to assess the combined effect of all exposures, we observed no overall association with KBC scores. This suggests a nuanced interplay between these factors. This study provides crucial insights into the complex relationship between prenatal exposures and child neurodevelopment. It highlights that MHG, DHA, and selenium can have differential and often sex-specific impacts on cognitive development, particularly in males. While individual associations were found, the overall combined effect of these exposures was not significant, suggesting that the interplay is highly complex. Ultimately, these results reinforce the need for pregnant individuals to carefully balance the risks and benefits of fish consumption to support optimal child neurodevelopment. Up next, our sixth paper out of 10. Anterior cingulate neurons display subregion-specific interaction with frontal eye fields revealed by anti/orthodromic stimulation and resting state imaging by Sahan Babapour-Farrakhan and Stefan Everling from Division of Neuropsychiatry and Neuromodulation, Department of Psychiatry, Massachusetts General Hospital, Harvard Medical School, Boston, Massachusetts, USA. This paper was selected because it provides a fundamental advancement in understanding complex brain circuitry by combining cutting-edge electrophysiological recordings with resting state fMRI, offering novel insights into the subregion-specific interactions between the anterior cing. Let's break down its key contributions. The anterior cingulate cortex, or ACC, plays a crucial role in controlling eye movements, specifically saccades, through its interaction with the frontal eye fields, or FEF. However, the precise mechanisms of this interaction have remained largely unknown. While imaging studies hinted at this connection, direct electrophysiological evidence was scarce. This study aimed to bridge that gap and shed light on this important neural pathway. To investigate this, researchers employed a sophisticated approach using macaque monkeys. They combined resting state fMRI with single-unit electrophysiology while the monkeys performed various cognitive saccade tasks. Crucially, they used anti- and orthodromic stimulation to precisely identify neurons in the ACC that were directly connected to or received input from the FEF, allowing for a detailed examination of these neural circuits. The findings revealed that ACC neurons primarily connected with the medial FEF, exhibiting significantly stronger positive functional connectivity compared to the lateral FEF. Interestingly, the lateral FEF showed a greater correlation between its task selectivity and positive functional connectivity with the ACC, particularly during the post-saccadic and reward periods of the tasks. Furthermore, the researchers observed that both mono and polysynaptic connections were associated with positive functional connectivity, providing a deeper understanding of the underlying neural architecture. This research significantly advances our understanding of how the anterior cingulate cortex and frontal eye fields interact to control eye movements, especially highlighting the role of the medial FEF. The detailed electrophysiological insights into synaptic connections and their relationship to functional connectivity are crucial for building more accurate models of brain function. These findings could pave the way for future investigations into how disruptions in these circuits contribute to conditions involving impaired eye movements, ultimately informing potential therapeutic strategies. Up next, our seventh paper out of 10. Metabolic reprogramming promotes AR-V7 splicing via SRSF2 lactylation in castration-resistant prostate cancer by Wenkai Zhu and Fei Xiao from the 5th Affiliated Hospital, Sun Yat-sen University, Zhuhai, China. This paper was selected because it uncovers a novel and critical molecular mechanism linking metabolic reprogramming to oncogenic splicing in castration-resistant prostate cancer, offering a significant advancement in understanding cancer progression and identifying potential. Let's break down its key contributions. Cancer cells often reprogram their metabolism, but we don't fully understand how these changes directly impact gene splicing, a process crucial for cancer development. This is especially important in castration-resistant prostate cancer, or CRPC, a very aggressive form of prostate cancer. CRPC is frequently driven by a specific variant of the androgen receptor, called AR-V7, which makes the cancer resistant to standard hormone therapies. Understanding the mechanisms behind AR-V7 production could unlock new treatment strategies. To investigate this, researchers utilized a combination of CRPC cell lines, patient samples, and in vivo tumor models. They employed advanced techniques like lactalomic profiling to identify proteins modified by lactate, alongside various functional and molecular biology assays. Their goal was to pinpoint specific molecular events linking metabolic changes to gene splicing. This comprehensive approach allowed them to uncover a novel mechanism driving CRPC progression. The study revealed several critical findings. They observed that CRPC cells exhibited significantly enhanced glycolysis, leading to elevated lactate levels and increased global protein lactylation. Crucially, they identified the splicing factor SRSF2 as a key target, specifically lactylated at a conserved lysine residue, K36. This lactylation was found to compete with ubiquitination, thereby stabilizing SRSF2. Ultimately, this stabilized SRSF2 promoted the alternative splicing of the androgen receptor into the AR-V7 variant, a major driver of CRPC. Furthermore, high expression of both LDHA and SRSF2 correlated with poor patient prognosis, underscoring their clinical relevance. This research provides a groundbreaking understanding of how metabolic changes directly influence gene splicing in cancer. By establishing a clear link between glycolysis, SRSF2 lactylation, and AR-V7 production, this study has identified a novel LDHA/SRSF2/AR-V7 axis as a promising therapeutic target for castration-resistant prostate cancer. While these findings are exciting, future work will focus on validating these mechanisms in larger patient populations and developing specific drugs to target this pathway, potentially offering new hope for CRPC patients. Up next, our 8th paper out of 10, antimicrobial peptide, WK13-3D, inhibits triple-negative breast cancer by blocking autophagic flux through inhibiting the AKT/mTOR pathway and targeting binding immunoglobulin protein, BIP, by Xuhong Su and Xiuqing Wang from College of Laboratory Medicine, Ningxia Medical University, Yinchuan, China. This paper was selected because it identifies a novel antimicrobial peptide, WK13-3D, and elucidates its mechanism of action against triple-negative breast cancer, a highly aggressive, and let's break down its key contributions. Triple-negative breast cancer is a particularly aggressive form of cancer, and we urgently need new and effective treatments. This study aimed to explore a novel antimicrobial peptide, WK13-3D, and its potential to fight TNBC. We specifically wanted to understand how it works at a molecular level, focusing on its impact on cellular processes like autophagy and key signaling pathways. To investigate WK13-3D's effects, we used both in vitro TNBC cell lines and an in vivo mouse model. We employed a range of techniques, from cell viability assays to advanced microscopy and Western blotting, to meticulously examine its impact on cell growth and molecular pathways. We also performed specific experiments to understand how it affects autophagy and key signaling cascades. Our results showed that WK13-3D significantly suppressed the growth and clonogenicity of TNBC cells. Mechanistically, we observed a clear blockade of autophagic flux, indicated by an increased LC3-2/I ratio and impaired P62 degradation. This peptide also inhibited the AKT/mTOR signaling pathway and directly bound to the protein BIP, leading to cellular stress. Importantly, these effects translated to anti-tumor activity in our in vivo mouse models. This study highlights WK13-3D as a promising new candidate for treating triple-negative breast cancer. Its unique mechanism of action, involving the disruption of autophagic flux through dual targeting of the AKT/mTOR pathway and BIP, offers a novel therapeutic strategy. These mechanistic insights are crucial for developing targeted therapies. Moving forward, further preclinical studies are essential to fully assess its potential and pave the way for future clinical development. Up next, our 9th paper out of 10. An oncostatin M receptor and chloride intracellular channel 1 crosstalk drives key oncogenic pathways in glioblastoma by Amir Hossein Mansorbadi and Arizu Jahani ASL from Department of Cellular and Molecular Medicine, Faculty of Medicine, University of Ottawa, Ontario, Canada. This paper was selected as the most impactful because it uncovers a novel and critical molecular crosstalk mechanism driving the progression and therapy resistance of glioblastoma, one of the most aggressive and intractable human cancers. Thereby, let's break down its key contributions. Glioblastoma is a highly aggressive brain cancer with limited treatment options. The oncostatin M receptor, or OSMR, is known to play a role in its progression and resistance to therapy. However, the exact molecular mechanisms driving OSMR's multifaceted functions in this devastating disease have remained largely unknown, presenting a critical gap in our understanding. To unravel these mechanisms, researchers employed advanced techniques, including mammalian membrane 2-hybrid high-throughput screening, to map the OSMR interactome. They also utilized genetic deletion studies, whole-cell patch clamp recordings, and a specific monoclonal antibody. This comprehensive approach allowed them to identify novel protein interactions and functional pathways crucial for glioblastoma. The study successfully identified chloride intracellular channel 1, or CLIC1, as a key interactor, physically associating with OSMR and EGFR-VII. Genetic deletion of CLIC1 dramatically disrupted this interaction, impaired STAT3 activation, and significantly slowed glioblastoma progression. Furthermore, they established that a distinct CLIC1-mediated current is indispensable for sustaining oncogenic signaling, highlighting a critical bidirectional crosstalk. This groundbreaking research reveals CLIC1 as a promising novel therapeutic target for glioblastoma, offering a new strategy to disrupt the OSMR/EGFR-VII/STAT3 oncogenic axis. While these findings are exciting, further preclinical validation and clinical translation studies are essential to bring this discovery to patients. Ultimately, targeting TMCLIC1 with specific agents like TMCLIC1-OMAB could pave the way for innovative and effective glioblastoma treatments. Up next, our 10th paper out of 10, a positive feedback loop between BOC1 and IL-1 promotes the progression of HPV-negative head and neck squamous cell carcinoma by Zishanbai Zhang and Lin Shan from Department of Otolaryngology Head and Neck Surgery, Beijing Tongren Hospital, Capital Medical University, Key Laboratory of Otolaryngology Head and Neck Surgery, Ministry of Education, Capital Medical University, Dongjiao Minxiang Street, Beijing, China. This paper was selected as the top choice because it identifies a novel molecular mechanism, a positive feedback loop between BOC1 and IL-1 beta, that drives the progression of HPV-negative head, and let's break down its key contributions. HPV-negative head and neck squamous cell carcinoma, or HNSCC, presents a significant clinical challenge, often leading to a poorer prognosis than its HPV-positive counterpart. Despite this, the precise molecular mechanisms driving its aggressive progression have remained largely elusive. Our study aimed to shed light on this critical knowledge gap, specifically investigating the role of a protein called BOC1 in this disease. By uncovering these mechanisms, we hope to pave the way for more effective treatments. To investigate the role of BOC1, we employed a multifaceted approach. We utilized both in vitro HPV-negative HNSCC cell lines and in vivo tumor xenograft models to study cellular and tumor-level effects. Our techniques included detailed gene expression analysis, assays for cell proliferation and apoptosis, and advanced chromatin studies. We also examined human patient tumor samples to correlate our findings with clinical outcomes, providing a comprehensive view of this complex disease. Our findings revealed that BOC1 is a critical oncogenic regulator, significantly upregulated in HPV-negative HNSCC. We demonstrated that BOC1 actively promotes tumor progression by enhancing cell proliferation and inhibiting apoptosis. Mechanistically, BOC1 directly activates the transcription of IL-1B, establishing a positive feedback loop with IL-1 beta that sustains oncogenic signaling. Crucially, pharmacological disruption of this axis using the IL-1 receptor antagonist anakinra significantly attenuated tumor growth in both laboratory and animal models. Furthermore, we observed a strong clinical correlation, co-upregulation of BOC1, BRG1, and IL-1 beta was associated with reduced overall survival in patients. This research has significant clinical implications. We've identified the BOC1 IL-1 beta axis as a novel and critical driver of HPV-negative HNSCC, suggesting its potential as both a prognostic biomarker and a therapeutic target. The promising results with anakinra, an IL-1 receptor antagonist, highlight a potential new treatment strategy for this aggressive cancer. Moving forward, the next crucial step will be to translate these exciting preclinical findings into clinical trials to assess if IL-1 or blockade can truly improve outcomes for patients with HPV-negative HNSCC. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.